In this video, I want to introduce you to T cell and B cell receptors. These receptors are complex proteins, multi-polypeptide proteins interacting together in quaternary structure that are bound to the cell membrane of your T cells and your B cells. Now, on a single, let's say a single B cell, you're going to have thousands of copies of the exact same B cell receptor. Okay, we're going to think of it as if it's just one, but for a given cell, you don't just have one receptor, you'll have thousands of it. Same is true for your T cell. You have thousands of T cell receptors on a single cell, but they will all be exactly the same receptor. What do I mean by that? It turns out that the recognition region, the antigen binding region at the ends of your T cell receptors and B cell receptors are random patterns of amino acids. They're random. So the rest of the proteins, and there are multiple proteins involved, their primary structure is set genetically. Okay, right? The genes give the recipe for which amino acid comes after which amino acid in order. Okay, it forms the sequence of amino acids. But when it comes to the regions that are the antigen binding sites at the ends of these receptors, like you see in this image here, there's a really cool mechanism involved that actually allows them to be randomly generated. Now, that's the part that's going to bind to antigens. And so most B cell receptors won't interact with chemically, won't bind to, won't have an affinity for most antigens. But you're going to create, because there are literally billions of possible combinations, you're going to create binding sites that will eventually bind to virtually every possible combination of antigen out there, whether it's a polysaccharide antigen or a protein antigen. The idea is that we can randomly generate all these and use these receptors to recognize just about anything that exists. It's a really crazy thought. Imagine if you were cutting out puzzle pieces and the shape that you cut was random. And then you took that shape and you bought boxes and boxes of puzzles at Walmart, and you started taking that randomly cut puzzle piece and searching for one that happened to be an exact fit. Chances are eventually you'd find one that's an exact fit, but the majority of them would never fit at all. Make sense? Think that through. If it doesn't make sense, be sure you're reading carefully about this as well. So that's the random generation of the binding sites, the antigen binding sites on the B cell and T cell receptors. Now, how do we make sure then that by binding to those receptors, we don't stimulate an immune response against self-antigens, against bits and pieces of you and me? If those binding sites are generated randomly, aren't a bunch of them going to recognize proteins and polysaccharides that are on the surfaces of our cells? Then we'd have an autoimmune disorder, wouldn't we? We don't want to go down that road. So we have something called clonal deletion. The idea is that the T cells and the B cells are tested. Before they're allowed to be out there in the world of immunity, fighting for us, they're tested. And the way that they're tested is literally a parade of virtually every self-antigen possible. You can imagine how many millions, if not billions, of those we could create in our bodies. A parade of these are passed by these T cells and B cells, these immature T cells and B cells. And if any of them just so happens to bind to it, they're killed. All right, they're shot on sight and eliminated, because it's that specific binding between the B cell receptor antigen binding site and an antigen that's going to stimulate and initiate the entire acquired or adaptive immune response. We don't want to initiate an immune response against self-antigens, only against non-self-antigens. So this process of clonal deletion is essential to make sure that we can distinguish self from non-self. If clonal deletion is faulty and we can't distinguish self from non-self, we're likely to have some serious immune problems, including autoimmune disorders. Take a look at figures 8 and 9 in your Bowman textbook and make sure that clonal deletion makes sense to you before you move forward. Now, a related phenomenon is called clonal expansion. We're going to see that when a T cell or a B cell binds to one that's been matured, right? It's survived clonal deletion, and therefore its randomly generated antigen binding sites should not recognize any self-antigens. When it does recognize an antigen in a lymphoid organ, this usually happens in something like one of your lymph nodes, it's going to activate, and that means it's going to grow. It's going to divide by mitosis and make lots more of exactly the same T cell or B cell with exactly the same receptor and its randomly generated binding site, so that now instead of 1 in a million happening to have exactly the right puzzle piece to fit, you've created temporarily millions and millions of exactly the same one. You've increased your sensitivity and your ability to respond to this specific pathogen that you're fighting at that moment. Clonal expansion is a relatively temporary phenomenon. We don't want our T cells and B cells to be dominated by just a few randomly generated receptors. We want the diversity of receptors so we can recognize all kinds of pathogens in the future. Let me give you a visual of how that clonal expansion might take place. So imagine these are all, let's call these helper T cells. You're going to learn about those in the next video. And let's say these are helper T cells found in one of your lymph nodes. So feel along your neck and find one of your lymph nodes if you can. And recognize that in that lymph node right now you have millions, if not billions, of helper T cells, each with a randomly generated pattern on its T cell receptor antigen binding site. All right, so they're all slightly different in that sense. Now all of a sudden you get sick, or even before you get sick. Let's say that you inhale a cold virus, and that cold virus attaches to your nasal epithelium. It enters into some of your cells, begins replicating, and some of your dendritic cells that are embedded in that nasal epithelial tissue, that layer, and we talked about this in the innate immunity, samples a couple of these viruses, digests them. It's going to literally crawl its way to the nearest lymphatic vessel, could take a day or two, and then slowly but surely work its way along the lymph ducts, very, very slow, nothing like your circulatory system, until it hits the closest lymphoid organ. Let's say it hits that lymph node right there in your neck. It is going to present bits and pieces of those cold viruses, literally, to as many of these helper T cells as it can, and saying, does anybody recognize this? Right? My toll-like receptors said this didn't look like self, am I right? Do we need to mount a response against it? And let's say that a couple of them out of these millions upon millions of patterns happen to recognize a couple of the fragments of these cold viruses. Those 2 or 3 are then going to be activated by clonal expansion so that they grow and divide, and temporarily their T cell receptors dominate the pool of T cell receptors. And that's going to increase our sensitivity to that pathogen and our ability to respond to that specific pathogen in the short term, just for the next couple of weeks. After a few weeks, many of these will die off, and we'll see that some of them remain around a little bit longer in what we call immunological memory. And that's really the basis for why we typically don't get infected by the exact same pathogen more than once, because of that immunological memory. And we'll talk about that a little bit later. So think about T cell and B cell receptors. Think about clonal deletion and the random generation of those antigen binding sites, and how clonal deletion really is the power behind distinguishing self and non-self. And then how clonal expansion happens when mature T cells and B cells are exposed to antigen, presumably not self-antigen, because the ones that bind to self-antigen should be dead by now, and how they temporarily expand in order to increase our army for the short term against the current battle, the current pathogen that we're dealing with. Let me know if you have questions about some of this introductory material.